Hi everyone, thank you for joining. In this third unit of the semester, we're going to be looking at the development of different model organisms. And in this lecture, we explore the development of one of the most studied model organisms, the fruit fly, Drosophila melanogaster. For many of us, when we think of Drosophila, the first thought that comes to mind is what a nuisance they can be. Although Drosophila can be quite annoying, these organisms have been invaluable for expanding our understanding of development. They're very prolific, they're hardy, easy to breed, and small. So they're easy to rear in laboratories and a lot cheaper than mice. Another advantage of working with Drosophila is that we know a lot about their genetics. So the fruit fly continues to be widely used for many different studies that are relevant to humans, from the study of cellular morphogenesis to disease and even aging and behavior. Drosophila development from fertilization to adulthood takes approximately 9 days. Following fertilization, embryogenesis begins with the division of the nuclei in the syncytial blastoderm, and that's not shown in this image, by the way. The formation of the cell membranes, which gives rise to the cellular blastoderm, happens after the 13th cell division cycle. This is followed by the cell and the tissue movements of gastrulation and organogenesis. The embryo then hatches as a first instar larva, and then it goes through 2 molds to become a third instar larva. The third instar larva becomes a pupa, and the pupa undergoes metamorphosis to give rise to the adult. Let's begin by discussing the process of fertilization. In Drosophila, there is no fast or slow block to polyspermy, but the sperm can only enter the egg through a single location, which is shown here, and that is known as the micropyle. Only one sperm can enter at a time, so this is thought to be important for preventing polyspermy. In Drosophila, the sperm enters the egg intact, so there is no fusion of the sperm and egg plasma membranes. A really interesting thing about Drosophila is that the sperm they make can be very long. For example, in Drosophila melanogaster, the sperm are almost 2 millimeters long, and that's about the same length of the adult fly, and that's actually 300 times the size of a human sperm. Not only that, but there are species that actually have sperm that can be 58 millimeters long, and that's 20 times the length of the fly. In some cases, the entire sperm enters along with the flagella, as you can see here, but the flagella is eventually defecated. So at some point, the sperm ends up in the gut of the animal. Drosophila sperm can be stored for about 2 weeks, so that helps to ensure that sperm is available when the oocyte is ovulated. Unlike in sea urchin and mammals, the Drosophila egg is activated before fertilization. Recall that one of the functions of fertilization in mammals and in the sea urchin is the activation of the egg's metabolism to begin development. But in Drosophila, this happens during ovulation, which takes place a few minutes before fertilization. Nevertheless, calcium is still involved in the activation of the egg's metabolism. In Drosophila, calcium channels open as a result of the movement of the egg through a narrow orifice. Another really important event that occurs prior to fertilization is the specification of the axis of the body. And that's mainly what we'll be focusing on in this lecture. Now that we discussed the most important aspects of fertilization, let's look at some of the key events of early Drosophila development. As we have seen in previous lectures, Drosophila cleavage is superficial. What this means is that a single layer of cells will enclose a yolky center. Another peculiarity of Drosophila that you may recall is the fact that they form a syncytium. So the nuclei divide without cytokinesis. That means they're sharing the same cytoplasm. As you can see, at around the 10th cell division cycle, the nuclei have migrated to the periphery. But at this stage, the cell membranes have not formed yet. However, the rate of cell division has begun to slow down. The first cells to undergo the process of cellularization, which is the formation of cell membranes, are cells located at the posterior of the embryo, and these are known as pole cells. These cells will form the gametes of the fly. In this picture, the nuclei can still be seen dividing without cytokinesis, while the pole cells at the posterior have already acquired their cell membranes. As in the frog embryo, the first few cell division cycles in Drosophila happen very quickly due to the absence of gap phases. Take a look at the following video to observe the division of the nuclei within the syncytial blastoderm. Although cell membranes do not surround the nuclei until after the 13th cell division cycle, recall that each nucleus is associated with cytoskeletal proteins, and because of this, the nuclei occupy a defined space. So what you're seeing here are the chromosomes in orange, and in green, that's the actin cytoskeleton. These are not cell membranes. The nuclei, along with the cytoplasmic islands that they occupy, are known as energids. And recall that in a syncytial blastoderm, microtubules are also essential for making sure that the nuclei retain their position as they divide. After the 13th cell division cycle, the process of cellularization begins. This process, which involves both microtubules and actin filaments, begins with the invagination or folding of the membrane between each of the nuclei. The invagination of the membrane between the nuclei and the elongation of the nuclei is regulated by microtubules. Once the membrane furrow passes the level of the nuclei, actin begins to constrict the cells at the basal end, and eventually the cells pinch off, forming individual cells. Eventually, this process results in the formation of the cellular blastoderm, which is a layer of cells surrounding a yolky center. As I mentioned previously, the first few cell divisions in Drosophila occur without any growth phases. So the embryo basically goes through M phase and S phase, so mitosis and S phase, and there's not even cytokinesis since the nuclei are dividing without the formation of any cell membranes. The fastest rate of cell division is observed during the first 10 cell division cycles, but after that point, cell division begins to gradually slow down, and you can see that gap phases are introduced into the cell cycle. This stage, which is characterized by the slowing down of cell divisions, is known as the mid-blastula transition, and we've encountered that term before. Other changes that happen during the mid-blastula transition include the switch from maternal mRNAs to the zygote genome. So that means that the maternal mRNAs are degraded and the zygote genome begins to be transcribed. At this stage, the process of cellularization has also begun. Soon after the formation of the cellular blastoderm, the process of gastrulation begins. As you can see in Drosophila, the body plan is pretty much the same in the embryo as it is in the larva and the adult. There is a distinct head region, and that's followed by 3 thoracic segments and 8 abdominal segments. The body segments that characterize the larva and the adult fly first become apparent during gastrulation. One of the first events to occur is the invagination of the mesoderm, and this happens at what eventually becomes the ventral side of the embryo. So this invagination is known as the ventral furrow. In addition to the invagination of the mesoderm, the endoderm also invaginates both anteriorly and posteriorly. In the posterior, the pulse cells, the ones that will become the gametes, also invaginate along with the endoderm. During gastrulation, the ectoderm and mesoderm undergo a process known as convergent extension. This process involves the migration of the cells toward the midline to form the collection of cells that is known as the germ band. And the germ band is basically the cells that are going to give rise to the thoracic segments and the abdominal segments. As you can see, the germ band extends to the posterior and then it wraps around the dorsal side of the embryo so that the most posterior segment, which is abdominal segment 8, is actually located right under the head of the organism. Eventually, though, that segment will retract. These segments will move back so that the most posterior segment is located at the right location, the posterior of the animal. But as you can see, even when the germ band is fully extended, the segments are already beginning to become apparent. Take a look at the following videos which show the process of gastrulation in Drosophila. As you saw in the videos, segmentation occurs during gastrulation. Other key events that happen during this time are obviously the segregation of the germ layers, but also organogenesis. For example, in Drosophila and in other insects, the nervous system forms from the ventral ectoderm rather than from a dorsal neural tube as it does in vertebrates. During gastrulation, neural progenitor cells, which are shown here in blue, invaginate at each segment on the ventral side of the embryo.